FDA's approval of a biosimilar product is based on a comprehensive review of scientific evidence. The comparative analytical assessment represents a battery of analytical approaches to characterize and compare the proposed biosimilar to the reference product. These physicochemical and functional studies may include biological assays, binding assays, enzyme kinetics, and other approaches. The analytical tests applied are reliable and very precise methods that provide data on the structural and functional similarity of the proposed product to the reference product to support a conclusion that they are highly similar. A proposed biosimilar with highly similar structure and function to a reference product should behave like that reference product with respect to safety and effectiveness. Biosimilar manufacturers generate data about molecular properties, or attributes, of the proposed biosimilar and compare these properties to those of its reference product. The molecular properties are ranked by risk of impact to the product's activity, pharmacokinetics, and pharmacodynamics, PK, PD, safety, efficacy, or immunogenicity, to determine those that are important to demonstrate biosimilarity. Particular focus is placed on these critical properties based on the nature of the protein and other information, such as its mechanism of action. This extensive characterization of the reference product informs the standards against which a proposed biosimilar is evaluated. Manufacturers conduct a head-to-head comparison of the proposed biosimilar's properties against the reference product. Results should be within an appropriate limit, range, or distribution. The analytical studies are conducted to compare the structure and function of the biosimilar to the reference product to show high similarity between the two. Highly sensitive analytical methods allow manufacturers to measure molecular properties across multiple lots of both the proposed biosimilar and the reference product. Manufacturers often apply multiple complementary techniques or orthogonal methods to better analytically characterize the properties of each product and further support the high similarity, or more sensitively assess for potential differences between them. The comparison between a proposed biosimilar and the reference product also includes product-related variants, like aggregates, precursors, fragments, or other modified forms of the desired protein. These data are compared across multiple product lots and helps provide assurance about the safety, purity, and potency of the proposed biosimilar. A proposed biosimilar application also includes data about process-related substances from the manufacturing process which arise from cell substrates, cell culture components, or downstream processing steps, and how they and the manufacturing process will be carefully controlled. All of this data and information helps support a determination that the approval standard is met and that the biosimilar is as safe and effective as its reference product. Analytical studies aided by novel and state-of-the-art technologies generally have higher sensitivity than clinical studies. This comprehensive battery is most likely to detect if differences exist between a proposed biosimilar and its reference product. The results can also influence decisions about the type and amount of additional data needed to support a demonstration of biosimilarity, such as tailored clinical pharmacology or additional clinical studies. For these reasons, the comparative analytical assessment is foundational to biosimilar product development. The comparative analytical assessment is critical for demonstrating biosimilarity and meeting FDA's rigorous approval standards. These assessments show that the biosimilar is analytically similar to the reference product. In combination with other data required for approval, it helps provide confidence that it will behave clinically like the reference product, with the same treatment risks and benefits.